This experiment works on the analysis of the effect of different temperatures on the fermentation rate of Saccharomyces cerevisiae and carbon dioxide production. The hypothesis of our experiment is, if the temperature is altered from the optimal temperature rate and the rate of fermentation is calculated in respect to CO2 production, then the enzyme will denature, thus decreasing the rate of fermentation and CO2 generation. This research is conducted on the influence of altered temperatures on the fermentation rates and CO2 production of the yeast. Adding a brief introduction, yeasts, also called Saccharomyces cerevisiae, are unicellular fungi which are also known as baker's or brewer's yeast. Now, fermentation is an anaerobic mechanism that occurs in the cytoplasm for ATP production. The process of fermentation is highly temperature sensitive, with the optimal temperature ranging between 25 to 30 degrees. Moving ahead, let's now understand how the process of glycolysis is carried out and also about the applications of fermentation. Now, the fermentation process first initiates with glycolysis, which involves the breakdown of glucose molecules into pyruvate. Now, the pyruvate is further reduced to ethanol. Here, NADH plays an integral role as a reducing agent, and carbon dioxide gas is released as a byproduct. Now, fermentation plays a significant role in various industrial processes such as food production, biotechnology. It's also used in food and beverage production, in biofuel production, and also in the biochemical industry. The goal of this experiment is to understand the effect of temperature changes on yeast fermentation, which will provide insights into optimizing industrial operations. Now let's talk about the methods which are used to carry out this experiment. We are going to be learning about the preparation or the materials required and the experimental setup in the coming slides. So, for setting up this experiment, the materials that we need are 2% yeast and glucose solution, 15 conical test tubes with graduations, fat glass test tubes, water baths set to 35, 50 and 65 degrees Celsius, ice bath, pipettes and pipette pumps, test tube racks, distilled water and markers. Now let's talk about the experimental setup or the procedure used in the experiment. First, label 5 test tubes and 5 conical test tubes. Fill these test tubes with 9 ml of glucose and 6 ml of yeast, and take another 6th test tube which will serve as the negative control, adding 9 ml of glucose and 6 ml of water to this test tube. Now, to ensure equal suspension, make sure to spin the yeast before adding it. Fill the tubes to the top and then invert them. Put them in a rack and set up one of the tubes in ice bath and incubate the others at 35 degree, 50 degree, 65 degrees Celsius and one of them at room temperatures. Next, incubate them for 1 hour to allow the fermentation process to progress, and observe them after every 15 minutes. After an hour, use graduations to determine the CO2 production. A total of 2 trials were conducted over a duration of 2 weeks to increase statistical power and ensure accuracy, therefore reducing any chances of error. Now, there are 5 specific variations that are being controlled in the experiment. The 5 variations are temperature, CO2 production, time, concentration of yeast and glucose solutions, and volume of solutions. So this was our experimental method or the procedure that we used to carry out this experiment.
In the upcoming slides, I am going to discuss the result and interpretation of the experiment. As we conducted the 2 trials in week 1 and week 2, and we observed the data, and the data we gathered, I am going to discuss that whole data in these slides, and the combined data of the standard deviations and mean. Also, I am going to conclude the whole experiment in the upcoming slides. So here is the result of our week 1, in which we take the trial 1. Here we take the 5 test tubes at the different temperature to observe the production of CO2 at the different temperature. We conduct the readings after every 15 minutes for 1 hour, in which we observed that the highest production of CO2 was observed at the 65-degree temperature, followed by the 50-degree temperature, and further by the 35-degree temperature. Minimum production of CO2 was observed at the room temperature. However, there was no CO2 production at the 0-degree temperature till the 45 minutes, and after 45 minutes there was 0.5 ml of CO2 production was observed at the 0-degree temperature in week 1. Data for the week 2, in which we take the trial 2. Again, we place the 5 test tubes at the different temperature to observe the production of CO2 at the different temperature. In this experiment, we observed that the highest production of CO2 in the yeast fermentation was observed at the 50-degree temperature, which was followed by the 65-degree temperature and the 35-degree temperature. The most interesting things in this trial was that the CO2 production was same after 1 hour at the temperature 65 and 35. Moreover, the result of room temperature and 0 degrees Celsius temperature was same as in week 1 trial 1. There was no production of CO2 at the 0-degree temperature, and moreover there is a very less production of CO2 at the room temperature, which means that for the yeast fermentation these temperatures are not sufficient. This is the combined table of mean and standard deviation of weeks 1's observation, in which the values of mean and standard deviations both are increasing with the increasing temperature and with the passing of time, which indicates that the CO2 production is also increasing with the increasing temperature and with the passing of time in each test tube. So here is the data of week 2's observation, and this is the combined data of mean and standard deviation. In this table we can see that with the increasing temperature the mean and standard value are also increasing, which means that CO2 production is increasing with the increasing temperature and with the passing of time. To conclude the whole experiment, in trial 1, highest production of CO2 was observed at the 65-degree temperature, whereas in trial 2, CO2 produced more at the 50-degree temperature, which surpassed the 65-degree temperature. Moreover, very less amount of CO2 was observed at the room temperature in both trials, and no CO2 was produced at the 0-degree temperature in both trials. With the increasing temperature, the production of CO2 was also increasing in this experiment. So this experiment does not fully support our hypothesis. In this experiment, with the increasing temperature, the production of CO2 was increasing and the fermentation of yeast was also increasing. For the future research, it would be recommended that they should take the higher temperatures like 80 or 100-degree temperature to fully support their hypothesis, because it could be possible that if they take the higher temperature, it might be possible that the CO2 production will decrease at the 80-degree temperature, which further decrease the yeast fermentation.